human being we search for the cues for their aliments by chewing herbs, terms, roots, leaves, uh, barks of the plants. So here some examples are given where this plantains or glides which are used in the treatment of malaria, like we see where it is a ephedra, right? So this used as a heart stimulant and a diaphoretic agent and for reducing cough. Yeah. And opium papaya juice was used as allergic and opium pills were used to treat cough, mental disorders, and aches and pains. Yeah. And this is the roots of Raulkia. Serpentina was used for treatment of hypertension and insomnia and insanity. So these are the some natural products which are used as a medicines. And the bark of the cinchona tree was used. No, no, no. The bark of cinchona tree was used for chills and fevers here, and it contains quinine and an anti-malarial drug. The drug discovery at present. So present is here the drug discovery. We have two options where the drug discovery uh, with lead and without lead, right? So nowadays, generally the drug discovery is applied by following the two methods, the drug discovery without lead and the drug discovery with lead. So present day drug development approach is mainly target oriented, and due to the rapid advancements in the biological sciences, so the better understanding of the geological mechanism at the cellular and molecular level is possible. Now, in most of the cases, target-oriented approach requires the identification of the target for a particular disease. So, understanding its structure and mechanism. So, as a result of developing in the synthetic methodologies, separation methods, and biochemical techniques, a more rational approach to drug discovery and design is possible today. So, in general, clinically used drugs are not discovered as such. Initially, a lead compound is discovered which may have desired biological and pharmacological activity, but may have other undesired biological activity or the toxicity. It may have solubility metabolism, or other problems associated with it. So therefore, the structure of the lead compound is modified by synthesis to enhance the activity and elimination of unwanted properties. In general, clinically used drugs are not discovered as such. Initially, a lead compound is discovered, uh, which is the smallest molecule here. Lead nothing but where the molecule, which is the smallest molecule, which is going to develop a drug compound here. So, which may have desired biological or pharmacological activity, but may have other the undesired biological activity. So it may have solubility, metabolism, or other problems associated with this. Therefore, the structure of the lead compound is modified by synthesis. Synthesis to enhance the activity or the elimination of unwanted properties here. Drug discovery without lead. So the approach we have two approach here with lead and without lead. So generally, the drugs are discovered based on the information derived from leads. So very few drugs are discovered without lead, just by accidental finding. We call it is this is the serendipity. So, for example, here you see the penicillin, which is a antibiotic, which was discovered by Alexander Fleming in 1928 during his studies of Streptococcus aureus librium. First benzoquinone, sorry, first benzodiazepine uh, transpiller drug was discovered by Dr. Leo during his work on the benzoxidazepine. Both these molecules form the basis for many other pharmacologically important molecules in their respective branches. Discovery of penicillin. Yeah. So in 1928, Alexander Fleming, who noticed a green mold growing in a culture of Streptococcus aureus, uh, could lies the bacteria, and his observation found it lies the bacteria and his observations formed the basis for the discovery of penicillin. Fleming was not able to uh, reproduce the result. Another scientist, Dr. Ronald here, could reproduce this observation and found that very special conditions are required for the production of penicillin. Only some strains of the molds produce penicillin and other strains do not. The original mold was found to be Penicillium notatum, but it gives low yield of penicillin. So penicillin chrysogenum, which was cultured from a mold on a grapefruit, gave better yield of penicillin. Penicillin prevents the bacterial development. That means bacterial development, bacteriostatic here. It could be administered only after the production of penicillin in suitable form. Sir Howard Florey, Oxford University. Uh, he was revealed only in 1940. So this delay was due to the two reasons here: emergency of the sulfonilamide and bacterial and World War II. So this is the correct structure of this penicillins, which was illustrated by the Robert Robinson and where the Carl was. Yeah. So several different penicillins were discovered where by right. So substituting the various alkyl groups here, so we get a variety of penicillins like if R equal to benzox, phenox, sorry, phenylmethoxy. So it is penicillin 5, where the if it is benzyl group, then penicillin G. So discovery of Librum. So Librum was first benzodiazepine uh, transferring drug, which was discovered serendipitously during the research of Leo's. This project involves synthesis of benzooxadiazepines of gender structure, which is given here. This is the benzodiazepine. So this was aimed to synthesize by this Leo Sternbach here. They prepared many compounds by varying the R1, R2, X, and Y here, where R equal to CS2N, where it is a dialkyl methylene derivative, and R2 is a benzene group, phenyl group. The compound was identified as but transferring activity, but none of them showed the interesting result. Therefore, the project was abandoned. But after many years, during laboratory cleanup, a vial was found which was prepared by taking, no, so which was taking methyl amine as one of the starting material, right? So like where the methyl amine CS2 NH NH2 here. So which is found which have prepared by taking methylamine as one of the starting material, and which was not screened for the activity. Screened for the activity, and as a last effort, being clean up everything. So this compound was submitted for pharmacological testing. So unexpectedly, this compound gave promising results in primary screening. Uh, yeah. For the structural identification studies showed the structure to be benzodiazepine peroxide, not for phenazolene, phenazolene trioxide. So as means they assume the clinozolene trioxide, but the actual structure is a benzodiazepine peroxide here, and and the scheme for the synthesis involved is this one here. So this accidental discovery of lithium was the basis for development of many benzodiazepine derivatives as the new transfeeling drugs. Uh, Had the sample of Librum not been found in the laboratory cleanup and had it has been submitted for the pharmacological evaluation, so all of the negative pharmacological results would have been reported for the phenolene trioxide. Next, the drug discovery with lead. So without lead means we are not going to synthesizing the starting material. And the drug discovery with the lead means where by taking the starting material which is known structure, and it has some stages. Uh, these are the stages, very different stages in the drug discovery. And you said the broadly speaking, the various stages in the drug discovery. First, we have to choose a disease. This is a very fundamental and basic stage. Choosing a disease, then choosing a drug target, and identifying a bioassay, and finding a lead compound, and the isolation and identifying the lead compound, structure, determination of the lead, if it is structure is known, and structure activity relationships, SAR studies and the QSR studies, and improving the interaction with the target, improving the pharmacokinetic properties, and metabolic studies, toxicity studies, and designing the manufacturing process and clinical trials. So these are the various stages uh, which are involved in the drug discovery with the lead compound here. When you're choosing a disease, so first you have to choose a disease when we are going to design or discover or invent a new drug molecule here. So first you have to choose a disease and then choosing a drug target here. Then identifying the bioassay. Bioassay is to study a extensive study about the target here. And finding a lead compound. Lead compound means the molecule which are having the pharmacological biological activity, the smallest unit which are having the pharmacological and biological activity with some undesired product, undesired properties like toxicity and the side effects, all these things here. Then isolation and identifying the lead compound. So once the lead compound is identified, so how to isolate and identify the structure of that compound. So if it is available from the extracted from any sources like plants here, so you have to isolate that and use identify the structure of that compound. Structural identification, then structural determination of the lead. If it is not known, if the structure is known, then so it is easy to go with that. If the structure is not known, then you have to determine the structure of the compound by following various methods like spectral methods, quality and chemical methods here, and structure activity relationship studies. So to improve the activity, you have to go with some structural modifications. By modifying the structure, you have to design a efficient with improved activity drug molecule again. So where it involves in SAR studies or the PSR studies. So all these PSR studies and SAR studies, so they are guiding us to improve the activity of the existing drug compound and improving the interaction with the targets. So then you have to see that means the interaction also the plays important role for the drug activity, where like by following the Lipinski rule, we know that Lipinski rule 5. So by following some rules, you have to you have to uh, improve the interactions with the target here. Then improving the pharmacokinetic properties, where the pharmacokinetics in the drug chemistry, we have two branches here, two broad classes like pharmacokinetic studies and pharmacodynamic studies. So pharmacokinetic studies and pharmacodynamic studies, where the pharmacokinetic studies which deals with the interaction and the activity of the drug compound here, and pharmacodynamics which involves the journey of the drug. And next, the metabolic studies like ADME properties. Okay, so the drug compound which is going to uh, under examination. So it must satisfy all these properties like uh, ADME properties, absorption, distribution, metabolism, and excretion properties here. So metabolic studies. So we have first the drug compound which is under examination. So it must be uh, having the good absorption property, then the distribution property here, then the metabolic. So when it is reaching the target and it is when it is showing that particular action at the site of action, so it must convert into the harmless, the non-drug compound here. Okay, then so that you which involves metabolic studies, then the toxicity studies. So the drug compound must not show any side effects. That side effect generally we call it as toxicity, and are designing the manufacturing process here. So once the drug compound is satisfying all these properties, so you have to design the manufacturing processes here for the marketing here. Okay, so then manufacture. So you prepare or synthesize that, you manufacture that particular drug compound which is under investigation. Then you go for the clinical trials. So uh, the thing here. So where it is going to uh, means it is applied on the particular living organisms to check the its working mode of that. So that is the clinical trials. In these clinical trials, we have seven stages of the clinical trials. Clinical trial one, two, three, and clinical trial two, five, and seven. Okay, so after satisfying all these clinical trials, so finally it is going to marketing here. Means before that marketing, it is approval. After clinical trials, so it is going for the approval step. Approval authorities like Food and Drug Administration (FDA). Then finally, it is going for marketing. So these are the stages. The drug which is discovered with the lead compound here. So here the particular area drug where identifying the particular area of the drug research suitable drug targets we have to identify. So normal drug targets are receptor generally we have the nucleic acids, this enzymes, proteins. So all these are the targets. These are the targets here. For example, you see that angiotensin converting enzyme one. Okay, so angiotensin, where it is angiotensin, it is decapeptide. So it is going to convert into, means it gets hydrolyzed and it converts into a peptide like angiotensin 2, which is converted in the presence of angiotensin converting enzyme. Okay, ACE enzyme here. And identification of bioassay. Generally, a bioassay screen is a means of determining in a biological system whether the compound under consideration has the desired activity or not. So that relevant reference compound. It also gives as a indication about the potency of the drug. That means the bias is a system which helps in testing the drug. So identification of suitable test system is therefore it is very important for the process success of the project involved drug development. Since the human testing is not possible at early stage of development, so normally the two stages of test are done primarily. One is in vivo test and in vitro test. Generally, in vitro gets two methods where in vitro is which are carried out on the isolated system tissues. Enzymes are the receptors outside and to the test the broad branch dealing the activities given drug molecule. Uh, the tracheal smooth muscle is isolated and the effect of that subjected drug material in manufacturing inhibition of its control in the test. In vivo tests are generally carried out in the experimental animals. So this test involves in the creation of observable disease symptoms in the animal, then treating it with drug and the anti-inflammatory effects it given, right? So inflammation is initially induced in the animal which are treated with the drug. So the random screening approach, all types of compounds are tested in the bioassay, so without considering their structural features. So the random screening is the zigzag screening here, where the random screening approach. So all the types of compounds So all types of compounds are tested in the bioassay. So without considering their structural features. So this approach was followed prior to the discovery of sulfur drugs. Nowadays, so it is not used very often. It is still very important to discover drugs or new leads that have unexpected and unusual success for various targets and unexpected activities. In 1940s, where the random spin of soil and the samples two important antibiotics, streptomycin and the tetracyclines here. But these are invented by the random screening. Pharmaceutical companies in such antibiotics result in the finding of many new leads to important antibiotics like streptomycin and tetracyclines. And another example, random screen is with the compounds for the cancer therapy. So all new compounds that were submitted were screened in a mouse tumor biopsy, and for these studies, some new anti-cancer drugs were discovered. Two major classes of materials, natural products and synthetic chemicals were screened by this method, non-random screening. This is a slight narrow approach that random screening. In this type, compounds having vague resemblance to weakly active compounds uncovered in a random scheme for the compounds containing different functional groups that lead compounds may be tested selectively. So finding lead compounds. So a lead compound is a compound which shows the desired pharmacological activity, pharmacological activity and as a starting point for the drug development project. The level of activity of the compound, lead compound may be lesser, it may have the undesirable side effects. So, tuning the structure of this, tuning the structure of this lead to get more potent with lesser side effects, constitute the major area of drug development. There are some important ways to discover the lead compounds for any drug discovery program. They are screening of natural products, medicinal products, screening of synthetic drugs, and existing drugs. So these are the some important ways to look for the lead compound. Uh, is the screening of natural products here, medicinal products like the ancient, traditional, okay, tribal drugs, and screening of synthetic drugs, existing drugs, and starting from the extracting from the natural ligands here in the complicated synthesis, multivariate synthesis, complicated the and limited prepared mind. So speaking of natural products, so we have yes, many compounds isolated from variety of nuts, natural sources, and biological active, and are used to direct science as medicines. Taxol, anti-cancer drug, and antihistamine, but they importantly for the development of new medicines. In fact, ancient civilization throughout the world mainly depends on local flora and fauna, full protein medicines here for their survival. Natural products provide a new and extremely complex structures. This complexity sometimes makes their synthesis very difficult, and designing simpler analogs becomes the essential approach. Some important examples of lead from plant and animal sources are briefly present in this discussion. Leads from the plant sources. So these are the morphine, cocaine, nicotine from tobacco, here, quinine from the cinchona bark. So all these are the uh, okay, the natural products extracted from the uh, natural sources. And the microbial tests are like fungus, molds, and the bacterium produces some uh, other may also have listening to treat microbial infections in human beings also. In fact, the observation for the insulin radio, the variety of particles as antibacterial agents. So extensive science research programs have discovered vast array of antibiotics such as very potent life saving threats. Some microbial metabolites also provide lead compounds in other fields. For example, from the microbial fermentation, uh, that like Aspergillus was isolated, which was shown to be novel antagonist of uh, yes, cholesterol, a peptide hormone. So this served as a starting point for the Merak scientists to develop very specific and potent non-peptide antagonist HCCK, A and CCK, B receptors. The fungal metabolites, lovastatin became the lead for drug which lowers cholesterol levels. And these are the examples you can see where the flavonoic acid cephalosporins and penicillin plant. So these are the investigated lead compounds and chlorophenicol, which is extracted from this Streptomyces venezuela, erythromycin, a metabolite produced by Streptococcus erythraeus. So these are obtained from the microbes here. Then venoms and toxins. A variety of venoms and toxins are used by animals as protection or to paralyze their prey. Some are extremely potent by reacting with the receptor of enzyme that require only the minor doses. So these compounds serve as starting points for the investigation of hormone receptors. Ion channels and enzymes such nasocodecs have provided important leads to develop muscle relaxants and hemostat, hemostatics. For example, we see that Aphrodite, which was isolated from the venom of become the lead for the development of antihypertensives, captopril and that is silazapine. and some other like drugs which are extracted from venoms and toxins. You can see the listed here. Now you see from these marine sources. Here is a pyrazine. Okay, so marine life generally often vast resource for need its need to so explorations, corals and sponges produce biological potent chemical with interesting inflammatory, antiviral, and anti-cancer activity. Like Curacin A, so which is isolated from marine cyanobacterium, has been shown a potent anti-tumor activity. And from the animal sources, we have the epi, epiptabidine, anti-analgesic drug, uh, epibacterium here, and medicinal folklores. So this is very important thing that the medicinal folklore medicines, that is a tribal medicines, herbal medicines, Ayurvedic drugs, so which plays important role in the identification of pharmacologically active natural products. So these can be used as leads for the development of new and potent medicines. For example, you see anthraquinones isolated from the uh, rhubarb roots used as a and have been lead compound for using acceptive and something. And some other important lead compounds are isolated from chlorpromazines. Here is the reserpine, antihypertensive drug, and the morphine. So morphine, it is opium drug, and screening synthetic banks. Synthesis of variety of compounds generally carried out by the pharmaceutical companies and research laboratories worldwide. So all the compounds that are synthesized are not biologically evaluated or not successful to reach the market. However, they are uh, reserved in synthetic compound banks here. Normally, pharmaceutical companies so often screen their bank of compounds whenever a new target is studied. Some of these compounds, which were synthesized either in synthetic research study or as intermediates, may show useful biological activity. For example, you see isoniazid, which is an anti-tuberculotic drug, which was developed from the screening of synthetic brands. We had one fluorophenol, 7 pyridine for phenol is the pharmaceutical antiviral here, which was developed from the studies of synthetic drugs. So these are the two drugs, isoniazid, anti-viral drug, and anti-viral drug here. Next, existing drugs as lead compounds, generally we call me too drugs. These are the me too drugs here. So, established drugs are normally used as leads by many companies. So, these drugs are useful to avoid uh, patent restrictions. The aim is to modify the structure of the drug to get a compound which may retain or may have improved activity. Existing drug may need improvement of to get a better dose from to improve the drug absorption duration, to increase the potency, to reduce the uh, daily dose, to avoid certain side effects. So these are the requirements. These are the aim targets to go for this its lead modifications to get a better dose from here. To improve drug absorption duration here, and to increase potency, the efficiency, to increase daily dose to avoid the certain side effects here. So, due to all these requirements, we go for these modifications. Sometimes these are labeled as me too drugs. However, most often they can often improvement over the original drugs. For example, modern day synthetic penicillins have the advantage over original penicillins as they are more selective, more potent, and more stable than the original penicillins. Antihypertensive drug captopril was used as lead for the development of the uh, lisinopril, enalapril, and sildenafil. Clinical observations during the clinical evaluation of the particular drug, other pharmacological activity like side effects in addition to the activity under study may be observed. So this observation is sometimes useful and serves as lead for the development uh, of another drug which different activity. For example, carbamide is an antibacterial agent and it was also found to have anti-diabetic side effects. So it is multiple functions. As such, it could not be used as anti-diabetic drug. So taking carbitamide as lead, carbitamide was discovered as the anti-diabetic drug, which is shown no antibacterial activity. Carbitamide is an antibacterial agent and also found to have anti-diabetic side effects. Similarly, promethazine is an antihistamine, which is sedative side effect, and enhancement of these side effects by altering the structures results in the development of chlorpromazine, which is used as neuroleptic agents. Next, the drug metabolic studies. Uh, in drug metabolism studies, the metabolites obtained from the drug in vivo are isolated and screened to determine whether the observed activity of drug is the original compound or its metabolite activity. For example, antibacterial agents Prantocil did not show activity against bacteria in vivo, in vitro, but it was found to be active in vivo. The activity was due to its metabolite sulfamide. Therefore, in the later studies, sulfamide was taken as a lead, and many sulfamides antibacterial agents were developed. So the anti-inflammatory drug, surinadic, was not active, but it is metabolite was known to be the respiratory activities which lead for the further development. Okay, here is this is the plant person. So it is product for this polymer, and due to the some reaction, this bond and is going to cleave and it is forming the sulfamide. And Sardat also says, and enzyme products have leads. Enzyme catalyze biochemical reactions in both directions in the body. And a product derived from the enzyme catalyzed reactions can serve as lead for designing enzyme inhibitors. That enzyme carboxypeptidase leaves the terminal amino acids of the polypeptide from the carboxylic acid based on benzene acid was designed to bind to the carboxypeptidase and it results in inhibition. So this observation also lead to the designing of capital. The nascent ligands and modulators are starting points here. So nascent neurotransmitters and other nascent ligands bind to the various and they are responsible for physiological reactions in the body. These systems may be useful as drugs. For example, neurotransmitters adrenaline and noradrenaline are responsible for the smooth muscle contraction in vascular smooth muscle and bronchial smooth muscle here. So therefore, by taking the neurotransmitters leads, beta adrenergic agonists such as solbitamol, dobutamol, dobutamol and Zamatirol were developed, which were helpful in the treatment of asthma and cardiac shock. Similarly, taking histamine as lead, histamine H2 receptor antagonist such as CBTD was designed. And you see, you take another example, which was development of the adrenergic antagonist potential, that is a CBTD. The neurotransmitter hydroxy uh, tryptamine was due to the development of sumatriptan, adrenaline, and noradrenaline was taken as the leads for the development of solvitamol. There are some modulators, leads, and nasal substance leads. So these are some the molecules, the strategies which are and the computer-aided drug design. Very nowadays, this field, uh, very fast-growing field here. So, if the detailed structure of the target receptor binding site is known, the possible leads that bind to the site uh, can be identified. This is possible if the target enzyme or receptor can be isolated and distanced. The extracrystallographic studies of this the structure and the binding site is identified. So by molecular modeling software program, designing the new molecule that fit in the binding site can be carried out. If a particular protein under consideration cannot be crystallized, the data from analog proteins can be used to develop new leads to or the drugs here. Next, the concepts of receptor This way, we have variety of the targets here. Like, okay, so here with the identification of leads for the possible drug molecule and the synthesis of new pharmacologically active compounds, studying the biological activities, studying and understanding their interactions with the different kinds of receptors. So means here the receptors, yeah, right. So receptors, means we have variety of receptors like proteins, enzymes, DNA, RNA. So nucleic acids, yeah, DNA, RNA, these are the nucleic acid. They are also acting as a receptors here. And determination of the drug absorption studies, distribution properties here. Okay, like the ADME properties. Okay, so we to say it is the ADME properties and study of metabolic transformation of these therapeutic chemicals and toxicity studies here. Right. So here, this all these extensive study about all these sections is takes place in this particular topic here. And you see the drug discovery in past. In past means like in the olden days there is no much technology, science and technology was developed here. In that case, so here the generally the peoples are curing their diseases by very variety of methods like chewing gums, herbs, roots by using the plants here, leaves. So all these are there. Some medicinal, what is that? That traditional medicines, or I can say it is the Ayurveda, sometimes kind of. Right. So here some plants which are having the medicinal importance is given like Shang Sha, which is the Chinese medicine here. So Chinese medicine. So roots of uh, Dicora fabricula. So it is used for fevers. So this plant contains alkaloids. You know that alkaloids. Okay, so the natural products we have the variety of classes like alkaloids, terpenoids, steroids, carotenoids. Okay, so all these are the class of the natural products. So this particular plant, this Chinese medicine, which is having the alkaloids, and it is used to treat for malaria disease here, and ephedra sinica, which is used as a heart stimulant, and it is also acts as a diaphoretic agent. Means diaphoretic agent means pers- perspiration producer here, and for reducing cough reducer also. It is acting as a cough reducer also. And some opium papaya contains morphine and codeine and many other alkaloids. Some opium drugs like morphine, okay, and codeine. So morphine, codeine. So the difference only thing is in the morphine we have the hydroxy group, and in the codeine we have the methoxy group. That is the difference here. So both are it's isomer, which is differs with the one methyl group here. Okay, and coca leaves. This was used as stimulant and euphoric agent here, and it contains a cocaine. This is also opium drug, and bark of cinchona we used for the uh, chills and fevers here. So cinchona abscinarum, okay, so which is acts as a anti-malarial drug, which is having the core structure is benign here. So now, structure activity relationship studies, SAR studies here. So here the biologically active compounds was generally first isolated from the natural sources. Once the structure of these compounds is established, the medicinal chemist tries to study the relation of structure to this activity. So usually physiological functions of a molecule is a function of its chemical structure. The aim of structure activity relationship studies is to identifying which parts of the molecule are responsible for the activity here. So once the essential basis skeleton is identified, a number of molecules are produced by varying the structure slightly without disturbing the pharmacophore, and their effect on the biological activity is studied. Drugs can be classified into two types, where the structurally specific drugs and structurally non-specific drugs. And structurally specific drugs means these drugs acts as the specific site and receptor or the enzyme. Their activity and potency are very specific to small changes in clinical structure, chemical structures. Yeah. And they generally have common structural features for similar biological activity. Normally, part of the molecule is associated with the activity. There is a penicillin. We have the types of penicillins, where by changing the R group here, uh, okay, phenyl methoxyl, here is a penicillin 5. And if R equal to benzyl group, then penicillin G. And chlorophyll, we have this chlorophyll structure, quantacil. So structurally non-specific drugs, these have no specific site of action and usually have no low potency. Similar biological activity may occur with variety of structures. Gaseous anesthetics, sedatives and hypnotics, many antiseptics and disinfectants. So even though only part of the molecule is associated with the activity, so multitude of modifications may be possible. Early studies simply involved. So synthesis of as many analogs as possible of the compound, testing these compounds to determine the effects of structural activity. After accumulating sufficient data, drawing conclusions regarding the structural activities, the binding role of the functional groups. So it was mentioned in the previous section that the drug molecules interacts with a particular receptor in the body by way of the functional groups in its results, the corresponding physiological reaction. So it is necessary to recognize these functional groups and determine which are very important for drug activity. Why? Because through many functional groups in the two, all of them may not have necessary for drug receptor bind. For this purpose, variety of compounds are synthesized in which one particular group of the molecule is removed or altered synthetically, systematically. So by studying the biological activity of these molecules, it is possible to find out which group are essential and which are not. Hydroxyl binding groups is interaction. The type of analogs that could be synthesized to establish their involvement in activities. Briefly, it is given here. First, the binding role of hydroxyl group. The common binding interaction of the hydroxyl group with the receptor is by hydrogen bonding. So conversion of hydroxyl group to methyl ether or an ester destroys or weakens such a bond. The hydrogen bond with the receptor is as given in this picture. Uh, Here was the drug and the electronegative atom. The hydrogen bonding is either lost or the considerably weakened by acylation. This is due to the both electronic factors and steric factor. To know the involvement of hydrogen bond of the hydroxyl group, so it can also be replaced by the CH3 as hydrogen in the synthesis of analogs. If the analogs thus prepared exhibits reduction in the activity or complete loss of activity, that means hydroxyl group is a essential for activity. Uh, Amphetamine, tyramine, and ephedrine have no effect for the adenoreceptors. This indicates so here the significant change in the properties of the mode. So this way, the planar carbonyl group is converted to tetrahedral alcohol, which results in the alteration in the geometry and the weakening of hydrogen bonding and dipole-dipole interactions. Carbonyl group may also be reduced completely to find out its role in the receptor binding and therefore in the activity. If the reduced analog shows decreased activity or loss of activity, it can be concluded that the carbonyl group is essential for the activity. The binding role of amino group. So amino group present in the drug molecule may be involved in drug receptor interactions via hydrogen bonding and ionic bonding. So the ionic bonding is more common with the amines. Converting amino group into amide results in the loss of this ionic bind interaction, which might ultimately results in the uh, disappearing activity or that loss of activity here. If this happens, we can conclude that availability is essential for this activity. And then the binding role of aromatic ring. Aromatic ring generally it is flat and they normally interacts with the flat hydrophobic regions of the receptors. So complete reduction of aromatic ring forms cyclohexane rings which are not flat and this results in a poor interaction. Therefore, To study the effect of aromatic rings on receptor binding, normally aromatic ring or reduced aromatic rings can be replaced by bulky alkyl groups here. So in such reduction results in diminishing it or the sorry lossing of activity. That means aromatic ring is that system is essential for this activity. The role of double bond also can be studied in the similar manner. The role of other functional groups, drug receptor binding, also can be analyzed by preparing the appropriate that particular analogs and study their activity. Next, the synthesis of analogs. Once it is established that a particular compound acts as a drug and its basic skeleton and the essential functional groups required for the biological activity or identified based on synthesis and synthetic analogies attempted. The natural compound acts as drug. Its potency may not be sufficient or it may have side effects here. Therefore, it is necessary to study analog mainly the to increase the activity here to get required potency to reduce the side effects because yes, very few drugs are ideal and do not have side effects here. And some of the drugs are having the side effects. So it is harmful to that particular living single organism. So you have to reduce that side effects and to prevent easy and efficient administration to the patients. Right. So the administration method, that administration process must be easy here. So in any drug development project, the medicinal chemist has these three objectives in the mind. One is to increase the activity, to reduce the side effects, to easy and efficient administration for administration route to this patient here. Drug development in the past was and sapphire. In this systematic development, uh, variations that can considered for lead modifications are so various substrates, chain extension or chain contractions, extension of structure, ring expansion, ring contractions, isolation and isostress, simplification of the structure, rigidification of the structure. So these are the main uh, what is that? So the objectives of the drug modifications studies. One is variation of the substance. Variation of substance means changing of the substance here. If methyl is there, you can change with ethyl or something else. And not that. So five member is there, six member. You can expand that structure, extension. Or like ethyl is there. You use the different like propyl, isopropyl like that. So chain extension. See if it is cyclic, right? Here the side chains are there. So you can extend the chain three member. For example, the side chain having three member, you can increase by adding two more carbons like five member. Sorry, three carbons with five carbons. Or you can contraction. If it is five carbons, you reduce to the two or three. So that kind of extension and reduction contractions and ring expansion and ring contractions. So ring contractions like increasing. Expansion, increasing of the ring. Five member. If it is existing one is a five member, you can increase with six member. Or contraction. If it is existing with the five member, you can reduce to the four member or six member. You can reduce to the five member. So like that. Okay, contractions means decreasing the size. It's a ring. Ring variations. So ring variations like if the heterocyclic ring is there, so you can, if it is oxygen carrying the oxygen here, the heteroatom oxygen is there. So you can see with this keeping the nitrogen here, the sulfur. So like that. Simplification of the structure. Okay. So if it is having the very complex structure, so you can simplify the structure here. Like morphine, you take morphine here. So it is a very rigid structure. So you can simplify and a rigidification of structure. Sometimes the complexity, okay, so simpler to complex, it is leads to increase the activity here. So all these are the objectives, all these are the objects for this lead modifications. So we'll see one by one here. So variation of substance. So after the identification of essential groups in the lead molecule, so other substances are varied to find out the influence of the activity, biological activity or the molecule depends on only the strong binding interactions such and these are certain other features such as density, lipophilicity, electronic distribution and size. So these are the main factors. So these properties can be varied by changing the droplet substance. What is that substance may be affects change in more than one physical features. Like you see one by taking one example, like replacement of methyl group of nitrogen uh, by other alkyl groups increase the basicity of nitrogen because the alkyl group it is not having the basicity, the nitrogen where it is basic nature. So if you replace the methyl with the nitrogen, your carbon, methyl carbon with nitrogen, so where the basicity is increasing and where the size also going to increase, yes. So wearing essential accessible substance is common method of finding tuning the fine tuning the binding interactions on the drug. So either of these changes may affect the activity which were known only after preparation studying more analogs and variation of alkyl substance here. Like you see, the molecules are having the such substance OH, NH2, OR, NHR, COSU. So these are the common substance which are used uh, in the drug, means that molecule here. So we can change with this the variety of substance. Okay, and so the changing of substance it is causing for better binding interaction with the receptor and extension of structure. So the strategies based on the assumption that extension of structure may result in the extra binding interactions, making the drug receptor binding more efficient. Okay, so extension of structure it is leading to the uh, providing the extra binding interactions with the drug receptor. In case of natural products like morphine painkiller, here is the extension of structure by the replacement of N-methyl group in morphine by phenylethyl group here, resulting in an analog which was found to have 14 times greater activity in the morphine than morphine here. So the phenyl group in the chain is believed to interact with the hydrophobic interaction with the receptor. So attachment of phenylethyl group uh, in other analogs result in the enhancement of activity. And this type of extension also observed in the like captopril, enalapril also, antihypertensive drug. So where the chain extension is cause of, in case of captopril, enhance the AC inhibitor activity as compared to captopril here, and chain extension and contraction. So these are some the objectives which is going to increase activity here. And very important terminology is isosters. Generally, the isosters which are the same activity, which are group. Okay, the where the groups are different, but showing the same activity here, where the molecular size, atomic weight is same. So you can replace one group with another here. So I'm stopping here.